Everyone. So beginning with the very first part of microbiology, which forms your foundation, and that is general microbiology. Now, we are going to start off with a lot of scientists, microscopes, stains, but do not take these topics lightly because, as I said, they are the building blocks. You learn these properly, not only will you get a lot of questions right, but you will also get a very strong foundation over the entire rest of microbiology that we are going to cover. So on that note, let me first introduce you to the world of general microbiology and tell you what all would you be covering under it. We are going to have scientists, stains and microscopes as the first part. Then we are going to go on and talk about all the different bacterial anatomical structures, shapes, physiology, all of this to do with bacteria. We continue with the genetics of bacteria. Since we're talking about them, we must know about the culture media. Then we wrap it up with sterilization and disinfection techniques, and a few miscellaneous topics shall go alongside each topic individually. Having said that, let's start right away, and let's start with the first topic, and that is the scientists related. So a very quick overview of all the scientists who've contributed. The first one whom we start with is Sir Louis Pasteur. Please remember, for every scientist, I've tried to make a mnemonic for all of you so that life becomes a little easier. So first and foremost, for Louis Pasteur, the way you have his name, that is his surname, that is going to tell you that after him, the Pasteur Institute in Paris is named. After him, and he is who gave us the pasteurization of milk, and everyone knows it's a mandatory thing to be done nowadays for all milk that is consumed. So Pasteur, Pasteur Institute, pasteurization, and Louis Pasteur tells us that which is the media that he has given us. He has given us the L for liquid media. So remember, now in most of these slides, you will see that there is going to be a small tiny little slide over here that will tell you that is your mnemonic section. Okay. So remember, the Pasteur is telling you number 1, Pasteur Institute in Paris, number 2, pasteurization of milk, and L is going to tell you that it is associated with the liquid culture media. So having said that, the story is not over. Do you know, apart from these three that you studied, Sir Louis Pasteur is said to be the father of microbiology. Now, there's quite a controversy around that because many people call him the father of bacteriology also. Having said that, he's definitely one of the fathers, of course. And the way you learn father is the way you are going to learn everything else that you have associated with him. So what do I mean by that? He is F for father. He's F for given us the fermentation principle. AT will tell you that he's given us the autoclave, which you know is a very, very important technique in sterilization and disinfection. And THER will tell you that he has given you two theories. There are two theories associated with him. One is said to be the theory of abiogenesis, which he said that there is nothing known as theory of abiogenesis. And he said that there is a theory which is there known as germ theory. So germ cell theory was approved and the abiogenesis theory was disapproved. So that is why I told you to learn it as we did with the mnemonic father. Let's do a recap. So F is going to tell me about the fermentation principle. AT tells us about autoclave. THER is going to tell me that one theory is disapproved, which is the abiogenesis theory, and the other is approved, that is the germ cell theory. Okay, now these contributions are not over. Do you know the first part of the mnemonic was Pasteur, Louis Pasteur. Louis Pasteur, whom you're calling as the father, and the father is going in a car. So remember, if you are asked what are the 3 vaccines which Sir Louis Pasteur has given, so he has given us the 3 famous car vaccines, means the cholera, anthrax and rabies. He has not identified these organisms. No, cholera organism has not been identified by him. The cholera vaccine has been given by him. So cholera, anthrax and rabies vaccine was given by Sir Louis Pasteur. So all in all, if you want to do a very quick recap of all the mnemonics, so first and foremost, the mnemonic is going to be as per his name, that is Louis Pasteur. The second mnemonic is that he's said to be the father, so he gave us fermentation principle, he gave us autoclave, and he was associated with a lot of theories. And father has gone in the car, so he's given us cholera, anthrax and rabies vaccine. So I think Louis Pasteur sir is now remembered by all. Now, because he was the father of microbiology, now let me introduce to you the father of modern microbiology. So again, all these fathers keep changing and you will find a discrepancy in many of the books while someone else is called as father of microbiology. But let me tell you, these father questions have stopped coming in the paper. So stop stressing over them, okay? Having said that, who is the father of modern microbiology? And that is Sir Robert Koch. Of course, he has a very Robert is a very modernized kind of a name, right? So he's said to be the father of modern microbiology. So what is the mnemonic for him? His name, of course, the name Robert Koch, that is going to be the mnemonic. So first and foremost, as soon as I say Koch, what are the Koch related things that come to your mind? Number one, there are Koch postulates, which I'm going to teach you right away. Then there is going to be Koch bacillus, which everyone knows is tuberculosis. TB is also introduced by him. So that is one thing. So K for everything, or KO to everything to do with Koch. He gave us Koch postulates, he gave us Koch bacillus. What did he give us with C and H? With C, he's given us three things. That is, so if you remember, Sir Louis Pasteur gave you the liquid culture media. So what culture media will the next scientist give you? C for culture media. That is, he gave us the solid culture media. Next, which organism has been given by him? The cholera organism has been given. So now that is why I made you revise this. Who gave us the cholera vaccine? The cholera vaccine was given by Sir Louis Pasteur. And who's given you the cholera organism? Sir Robert Koch has given you cholera organism. What is left with C? A color was also given. That is the aniline dye. So repeating once again, there is a cholera organism, there is a culture media which is solid, there is a color which is the aniline dye. And finally, what is with H? With H, he has given us the hanging drop motility. Yes, that has been introduced by him. So let me tell you what is hanging drop motility. We'll talk about that when we go to the bacterial anatomy part. But hanging drop motility is a test for motility. It is a test which we do for knowing whether an organism is motile or not. Well, on that note, that is over. But one thing that I want to teach you in detail are the Koch postulates. How many Koch postulates do we have? Now remember, we call it as 4 plus 1, because earlier there were 4 Koch postulates which are written in front of you. I am today going to add on one more Koch postulate which makes it a total of 5. So let me teach you the old ones first. Best ways, either you read it like this, and randomly reading microbiology is never going to get into our brain, right? So it's always better make tiny little mnemonics or tiny little diagrams. So first and foremost, what is the first part? Constant association of the causative organism with the disease. What does that mean? So for example, I give you a case of Mycobacterium TB. I give you one bacteria. That bacteria is Mycobacterium TB. What is it always going to cause? Can Mycobacterium TB suddenly end up straight away causing meningitis? No, the first thing that it will cause is tuberculosis. It's fact, it's fixed, right? That there's a constant association that Mycobacterium tuberculosis will cause tuberculosis. So whatever disease it is causing in the human, it should be the same disease all the time. There's a constant association. Point number 1. So now you have a patient who's having TB. Tell me, can you isolate TB in a culture media? From this human being, if I take a sample, can I grow it in a culture media? Yes, of course, like this, you can grow it. TB ke liye there's a culture media which I'll tell you later on. So, isolation should be possible in the culture media. That's another thing. Coming to the third one. Now that you've grown it into the culture media, from this culture media, if I take a little bit of the colony, so little bit of the colony from the culture media I took and I put it into an animal. So let's assume the favorite animal for all lab tests is that rat or the mouse. So you put it into an animal and it caused the same disease. So for example, if in humans it had caused TB, then in the animal also it should cause the same disease. It can't cause leprosy all of a sudden, okay? So remember, there is a constant association in the animals also where it is going to cause the same lesion. Now what do you do? From this animal, if I try to re-isolate, again grow it on a culture plate. So now from this rat or from this animal, if I try to grow it on a culture plate, I should be able to grow it once again. That wraps up your end of the 4 Koch postulates. So it's like a story. The first part is that every time it should cause the same disease, constant association. Once it has caused the disease, you should be able to grow it in a culture media. Once you've grown it in a culture media, you should be able to cause the same disease in the animal. And once you've caused it in the animal, you should be able to re-isolate it again in a culture media. So it's become like a nice story, but these were only the 4 Koch postulates. I am interested in the fifth one now, and the fifth one says that whenever there is an antigen, where will the antigen come from? Antigen is going to come from the organism. They are saying that the person should be able to produce antibodies in the serum. The human being should be able to produce antibodies. That is one thing that has been mentioned by now. So repeating, remember now how many Koch postulates? It's common sense, na? If there's an antigen coming in my body, if like COVID came into our body or any other streptococcus, a bacteria comes into our body, we make antibodies, right? So that postulate is pretty much very common sense. Coming all the way back, so Koch postulates were given by Sir Robert Koch. I can safely say now there are 5 Koch postulates. Okay, now what are the exceptions? Not all organisms. If you lay down rules, there will always be some people who will not follow the rules, right? So remember, not everyone is going to follow these, and LPG does not. What is LPG? Mycobacterium leprae does not follow it, because can you grow Mycobacterium leprae in any culture media? No. For Mycobacterium leprae, you need an animal, which I'll tell you need armadillo. So you cannot grow it in a culture media. Next is what? P for pallidum. Treponema pallidum doesn't follow these rules, and gonococci. There are many others also. I will tell you later on. There are organisms called rickettsia, chlamydia. They are also going to pose a problem. But abhi ke liye you have to remember LPG. Repeating once again, Mycobacteria lepra, Treponema pallidum, and gonococcus. They do not follow the Koch postulates. Okay, we are not over with the scientists. The next scientist whom we have is Sir Paul Ehrlich with their respective photos. So the mnemonic for Sir Paul Ehrlich is also Sir Paul Ehrlich. Everywhere, see, the mnemonic for Louis Pasteur was Louis Pasteur, Father Carr. The mnemonic for Robert Koch was Robert Koch. Now the mnemonic for Paul Ehrlich, obviously, which organism has he given us? The Ehrlich word is telling me that he's given us the Ehrlichia organism. Okay, what is he a father of? Again, so like I said, if you take the CH, you will get he's the father of chemotherapy. He gave us the entire concept of chemotherapy. So CH for chemotherapy, and Ehrlich will tell you Ehrlichia organism. Ehrlichia organism was a recent question that came in the previous year papers. Okay, next, remember P and A. He has received a prize, P for prize. He has received a prize, and that prize is a Nobel Prize for what? For toxin antitoxin standardization. So this is also a little bit of a common sense that if someone is, if you have a toxin, there will always be an antitoxin which can neutralize it. So he gave that concept that a toxin antitoxin can be neutralized, and that is what is for which he received a Nobel Prize. And what is A? What is the A over here after the Nobel Prize? He has received a Nobel Prize for acid fast stain. And actually it's a prize, not a Nobel Prize, but he did receive a prize for acid fast stain. So now is it becoming like a little bit of, am I contradicting myself in advance only? Because acid fast stain is also known as the Zeal-Nelson stain. Means it is the Zn stain. That basically means, guys, that now comes the real revolution time. That is, Ehrlich was actually the one who gave us acid fast stain. Today you know it as the Zn stain, but Zn was just the people who modified it. They modified it, and now today everyone knows it as the Zn stain. But who's the original inventor of the acid fast stain? It is going to be the Sir Paul Ehrlich. I hope you'll remember it. Quick recap. When we are talking about Sir Paul Ehrlich, first thing is the Ehrlichia organism. Next is he's the father of chemotherapy. P is going to be for the Nobel Prize, and A is going to be for acid fast. Coming to the next scientist, that is Sir Joseph Lister. Again, the name that is Joseph Lister is going to be your mnemonic, because what did Joseph Lister give you? So I learn it as Joseph because he is the father of antiseptic surgery. So Joseph is antiseptic surgery. What is antiseptic surgery? You all know that in today's date, once you are working in a you're working in any OT, any operation theater, you have to maintain an antiseptic environment. So he first used carbolic acid. What did he first use? He first used carbolic acid, and that is how he established that yes, I need to maintain an antiseptic environment, and that is very, very important. Having said that, the next one who needs no introduction, the father of light microscope, and that is Sir Anton von Leeuwenhoek. L for L because there are so many microscopes, so you should be knowing that the father of the light microscope is Sir Anton van Leeuwenhoek. And let me tell you, the very first thing that he saw, the very first thing that was visualized by Sir Anton von Leeuwenhoek was actually known or labeled as animalcules. Animalcules. So yes, having said that, Anton von Leeuwenhoek, L for L, light microscopy, animalcules. But I want to tell you, and I don't know how many of you have actually gone that much back ever in literature and wondered, so how many, when you have a microscope, I'll be showing you a microscope in a while, you look with both your eyes, right? Both your eyes can be put into a microscope. So it is going to be what? Binocular microscope. But actually, if I ask you, did Anton von Leeuwenhoek sir actually identify that? No. What Anton von Leeuwenhoek identified was a simple light microscope, the one which you could see with one eye, uniocular wala. So that is the simple light microscope. I'm just telling you a little bit of detail. There were the Janssen duo, the Janssen brothers. They are the ones who actually gave us the compound microscope. So the duo, two people, they actually gave us the two eyes wala microscope. So that is going to be the compound microscope. That I'm only telling you if you're aiming for little higher exams, right? Okay, moving on. Now we have light microscope done. So who gave us the electron microscope? That is going to be E for E. Ernst Ruska gave us the electron microscope. Like I said, Ernst Ruska gave us electron microscope. Leeuwenhoek gave us light microscope. Easy to learn. But there's another E that is known as Edward Jenner. So don't get confused. Edward Jenner did not give you the electron microscope. Edward Jenner, Word gave you the vaccine. Edward was given the award for the vaccine, the first vaccine. What was the first vaccine made for? The first vaccine was made for smallpox. And I know all of you are aware from PSM that smallpox is now eradicated, hai na? But why was it eradicated? Because we had such a nice vaccine that was available for it, and that is given by Sir Edward Jenner gave you the vaccine. And do you know smallpox vaccine, how was it prepared? The smallpox vaccine was prepared by using cowpox. Actually, it was prepared by using cowpox. Coming to the next scientist, KBM, 3 alphabets, Carrie B. Mullis, which means it gave you something to do with 3 alphabets, and that is polymerase chain reaction. I want all of you to recall your pathology lectures. Do you remember what was the gold standard of polymerase chain reaction? The gold standard methodology was what? That was Sanger sequencing, right? Now, I hope all of you will be able to also recall that I told you that Sanger was the, now when you go back to the pathology lectures, you will see that Sanger has the only one that has a G. That is how we had learned that it is going to be the gold standard, right? And also, if they ask you who gave you Sanger sequencing, so obviously the scientist Sanger gave us. So now read the question carefully. Both of them are PCR. When I write PCR, it's just overall in general PCR. So P, C and R are 3 alphabets. So Carrie B. Mullis is going to be 3 alphabets. On the other hand, Sanger is sequencing, so Sanger ne diya. Okay, the most common sense thing that I'm going to tell you now is who gave us the gram staining. So the scientist was only H.C. Gram, and H.C. Gram gave us the gram staining. Okay, the scientists are almost coming to an end. Okay, Klein-Berger, the L needs to be highlighted. What is Klein-Berger? L forms. What are L forms if you ask me? They are cell wall deficient forms. L forms are going to be cell wall deficient forms. Okay, what did Alexander Fleming, this you've been reading since your NEET UG days. What did Alexander Fleming actually come up with? So please remember, he gave us the very first dose of penicillin. Penicillin, this K used to come in your NEET UG, if you remember, we used to study it in our 11th and 12th high school, that it was basically penicillin, that is Alexander Fleming who gave us. Finally, the new one, Barbara McClintock. Barbara McClintock has given us something known as transposons. What are transposons? Transposons are referred to as jumping genes. Transposons are referred to as jumping genes. So repeating, Barbara McClintock, transposons or jumping genes. Now I'm going to tell you, during this era of COVID, when everything was in a state of lockdown, but one thing that went on was the giving of Nobel Prizes, and that is a must because recognition is always important. 2 very important Nobel Prizes were given, so hang out and listen to that. HCV, that is hepatitis C virus. Who has told us everything about hepatitis C virus? HCV, 3 things. So 3 people had to come together. Hulter, Alter, and Rice. Houghton, Alter, and Rice. These are the 3 scientists that have come together and have given us these. So obviously you don't have to know their pictures as such, but it was published in every paper, so I thought let me share it with you. But Houghton, Alter, and Rice, they gave us all the things and all the pathogenesis of hepatitis C. But what was even more important was who gave us CRISPR-Cas9, and there were 2 chemistry scientists, 2 female chemistry scientists who gave us CRISPR-Cas9, and they were Madame Carpentier and Madame Jennifer. So remember, Carpentier and Jennifer, I've just picked up the more easier names out of their names. So Carpentier and Jennifer were 2 actually chemistry scientists who were given the Nobel Prize again in the era of the lockdown. This is what happened. So they have given us CRISPR-Cas9. If you're aware of it, excellent. If not, don't worry. When I go to bacterial genetics, I'm going to explain to you what is CRISPR-Cas9. The same CRISPR-Cas9 I have explained in pathology genetics also, because it is being used by humans also, it is being used by bacteria also. So it's my duty to explain it to you in both places. Having said that, that brings us to a wrapping up of the first part of this particular series, and that is going to be the scientists. What is the next topic that we are going to go on to, which is based on our topics that we are doing? The next thing that we start off with are going to be the stains. So let's start right away. And obviously, when I talk about stains, I know immediately you want me to rush towards gram staining, which I'm not going to do. I'm going to first tell you all the basics and then go on to gram staining. So first and foremost, if you have to divide the stains, or if you have to talk about how a stain is done, I'm going to make a slide over here. Okay, let's make a slide over here. So for example, if this is the slide, and whatever sample, you've taken a sputum sample, or you've taken a CSF sample, or whichever sample has come from the patient, you are going to put your sample onto that slide. You know, after this, you are going to treat it with, you've seen, staining means you're going to treat it with colors, with alcohol, with water, with so many things all together. So before you start treating it with so many things, so many fluids, you want to make sure that this sample is sticking onto the slide. It's not going to get washed off. What if I put water in my sample? Patient sample only gets washed off. So please remember, you're going to put the sample and make sure that it is going to remain there only. How will you make sure? You will do a step called fixation. So either you are going to put it over a flame, you're just going to pass your slide over a flame, that is heat fixation, or in some cases you add a chemical to fix it, and in most of the cases that chemical is a kind of an alcohol, that is methanol. So simple. I want that, for example, if this pencil is my sample and this hand is the slide, I want that it should stick onto it. So what will I do? Either I will pass it over flame, I will just heat it for 2-3 seconds, or I will add a little bit of methanol. So this pencil has now become permanently stuck onto my slide, and that is what I wanted, because now I can add colors onto it. So let me add some colors. Let me begin with something very simple. What do we call as a simple stain? The first category of stains. Simple means one color. The entire slide is going to have one color. So either you have added methylene blue, so the entire slide will become blue. If you want to make everything red, you can add something like basic fuchsia, and everything is going to become red. So basically, you're not interested in making some things red, some things blue. No, you're interested that everything should have a single color, and that single color is either blue or red. So methylene blue or basic fuchsia, but that is not how it can work. If you're studying with me Path and Micro, you know for a fact that I'm going to not only make my notes very, very colorful, but the world of Path and Micro is only very colorful, right? So now I'm starting off with the boring things, but I'll ultimately make it very colorful. So right now, 1-1 stain, either all blue, all red. I'm going to make it more boring because now I'm going to make it a little black and white. The second type of stain that you have is negative stain. Negative, what does it remind you? Does it remind you of, I don't know, I might sound a little old as I say that. You might not have seen, but yes, when I was young, we did not have too many phones to click pictures with. So we used to take pictures with the cameras, and in the camera there used to be a reel. But I'm sure everyone has seen how those negatives were, those brownish blackish negatives from which you could develop more photos. I'm so sure you guys have also seen it in your childhood, maybe your parents used to use. So negative, what used to be a negative, if you remember? A negative used to be when the background was all dark. The background was all dark and you could see some person's random highlighted picture that when it will be printed, you'll see the proper picture out of it, right? Same thing I am doing over here. Am I going to stain the organism? No, I will stain the background. So, for example, if this is the background, I have given the entire background a black color. So I have made the entire background black, so that if this is the organism, will it shine out? It will shine out as white, right? So for example, if this is all black, it is going to shine out as white. That is what is negative staining. So this I'm going to tell you as an example of Cryptococcus, very famous example, and I'll directly write it out as what disease. What does Cryptococcus cause? It causes cryptococcal meningitis, right? It causes cryptococcal meningitis. So now everyone, guys, please note, cryptococcal meningitis is a capsulated organism. It has a capsule, but the problem is that if I want to stain that capsule doesn't take up any color. It's like this. It's not going to take up any color. How do I look at it under the microscope? I stain the background black. I stain the background black. So what did I do? I added something called India ink or nigrosin. These are two separate stains, and these are negative stains. So you can do, what have they done? They have stained which part of the slide? They have stained the background. The background has become black, and can you see the bacteria and the capsule, how the capsule has basically been highlighted out? And that is exactly what you need to know. So look at this picture also, which is shown over here. Can you see all the background is black, 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 and this is how the capsule has been highlighted. Well, having said that, the boring part, that is negative and black and white, is over. But remember, for cryptococcal meningitis, it is very, very important, because whenever meningitis means what sample, they will ask you that when we give a CSF sample, what are you going to do on a CSF sample? You do India ink or nigrosin. Now finally, moving a little towards color, but still in the black and white zone only, the third stain that we have is impregnation stain. It's very easy to read all these things written in the book, but basic concept nahi pata, then it's a waste. So let me first tell you the basic concept. For example, I give you something very, very thin, like something like a hair. Okay, my hair today are becoming quite important for teaching you stains. So very, very thin like a hair. Can you look at a hair, single hair, very easily? No, it's very thin. So if something is very thin, then you can't look at it under the microscope. You make it fat, you make it thick. So what do? If this is the thin hair which I can't see, I'm going to cover it with something, multiple layers. I'm going to make it wear lots and lots of clothes, so it's going to look big and bulky. That is what I do. Let me tell you some thin things. There are some thin structures like flagella, like spirochetes. These are very, very thin, in very few 0.0 something microns, very, very thin. So if you want to see it under a microscope like that, you can't. Now I'm going to make them fat. How am I going to make them fat? I am going to deposit silver stains. So if this is the flagella, I will deposit so much of silver around it. And do we already have one concept? If you've seen the patho lectures earlier, we already have a concept that silver is going to be what? Silver is always going to give a black color. So have a look at this picture, guys. Can you visualize that earlier these are actually spirochetes? Let me tell you, if you're wondering what exactly is being shown over here, these are actually spirochetes. And please remember, they were originally very, very thin, but here I'm able to see black structures. Why? Because I have made them mota. I have made them thick. How have I made them thick? I've deposited a lot of silver over them. That's the concept. When you are impregnating something, you are making something thicker by adding. So what am I adding over here? I'm adding a lot of silver. So this is particularly done for thin structures like flagella and spirochetes. Do you guys have any idea? If you've not studied bacteriology lectures, do you have any idea what exactly is a spirochete? Spirochete, for example, is Treponema pallidum. Just as an example, Treponema pallidum. There are many other spirochetes, but Treponema pallidum is the one that causes syphilis. So yes, for the diagnosis of syphilis, I will use this silver staining technique. And let me tell you, there are two things. Now, when I'm dealing with syphilis, syphilis usually comes to us as what? Syphilis comes to us as genital ulcers, okay? Syphilis patient presents as genital ulcers. So either I am going to get a little bit of fluid secretion from the ulcer, ye wala sample, or I'm going to get a tissue sample. So which kind of silver stain will I use? Both of them are silver stains. But if I get a tissue sample, I use the Livadetti stain. And if I get a fluid sample, I use the Fontana stain. How do I learn it? So see, for F, Fontana stain is used for fluid specimens. Repeating, F is Fontana, F is fluid. And Livadetti, TI is going to be used for TI tissue samples. That is the method of learning it. Repeating, Livadetti, TI tissue samples, Fontana, F fluid samples. Works well with everyone. Thin structure, made fat. How? By depositing silver. So we've done three things, and I've spoken a lot about very boring black and white things. The first one was simple. In simple, what did we have? That only one color is added, either all blue or all red. The second one was what? That these are negative stains. What are negative stains? Negative stains are going to be that the background is going to be stained and then the main organism is all highlighted out. What is the third one? The third one are the impregnation stains. What is impregnation? Impregnation basically means a thin structure has been made fatter. Now finally entering into the world of colors. Differential stain, the bread and butter of every microbiologist, gram staining. Then acid fast, then Albert. Let's start one by one. For gram staining, first I'll write down three things. For gram staining, please remember, you want to learn gram staining because you have to. It's the bread and butter, right? So what are you going to do? You are going to come in and stain. Come in and stain. Learn it yourself. Come in the lab and stain it. Okay, next, you want to learn Zn or acid fast. You are asking me for a mnemonic for Zn stain. Okay, class has asked mnemonic. Class has asked mnemonic. And finally, you are asking me for Albert stain. So Albert stain ke liye we are going to have Tim. So many students have a problem in learning which chemical comes in which of them, and now your life is going to become easier. So let me start one by one. So starting with the first one, and that is the gram stain. And like I just told you, the mnemonic for the gram stain is going to be come in and stain, right? Come in and stain. You want to learn it, you'll have to come in inside the lab and stain it. Means what? C is going to be for the first thing that is added, crystal violet. Now obviously guys, as I'll tell you the concept also, that when you're dealing with a gram stain, you're dealing with either gram positive or gram negative, right? So now having said that, when we say first thing added, crystal violet, obviously you can see everything has got a violet or a purple color. Now be it gram positive organisms, they are also purple, and gram negative organisms are also purple. So I've written PP for purple, okay? Gram positive organisms are also going to turn purple. Let's write it down to avoid any confusion. So gram positive is also purple, and then gram negative is also purple. So crystal violet is the C. What is the IN? The next thing added, you can see over here. IN added is the iodine. Now, why is iodine added? Because they ask you a question on iodine. Iodine is a mordant. You might have seen this in previous year questions. What is the function of iodine? You know what is a mordant? Mordant is a hammer. You've added, so what have you added? Right now, what have you added? You have added crystal violet, right? Crystal violet added. On top of that, you want crystal violet should stick over there. So you add one hammer on it, and that hammer is like mordant. That is iodine. What are you going to do next? Look at the next alphabet. Come in and. So now you're going to put A for decolorizer, that is alcohol. So the next thing that you add over here is going to be alcohol, or we can also use acetone, whatever we may be using, but that is actually going to be a decolorizer. Whose color are you going to remove? Whose color are you trying to remove? So after this step, I realized gram positive organisms were initially purple. They are still purple. I have not been able to remove their color, but suddenly I realized gram negative organisms have become colorless. Gram negative organisms, they've become totally off color, totally colorless. So now what do I do? I'll tell you the reason also, but what do I do? I can't leave them colorless. I will have to add one color onto them, right? So the last one is S, and that means, can you see a reddish color stain coming your way? Yes, so this is known as saffronin. It is known as saffronin red, which means that now we are going to get what color? So see, gram positive, ultimately I realized gram positive started their journey with being purple, until the end they have ended up being purple. Gram positive did not give up any on their stains, but gram negative organisms, finally I'm seeing they are being red or pink in color. Red or pink. So what is your final interpretation? First and everyone, please revise with me so that I can tell you what is the principle behind it. So what is it? Come in and stain. Can you tell me what all was used? The first one is crystal violet. Next is the hammer or the mordant, that is iodine. Third one is going to be alcohol or acetone. And fourth one is going to be saffronin red. And can you see the interpretation in front of you? What are all these purple organisms? These purple organisms are all gram positive. And what are all these reddish ones? The reddish ones are going to be gram negative. So simple interpretation is also learnt. Now obviously you will ask me, what are the variations, and which is, if I ask you out of these, which is the most important step? Come in and stain. Which is the most important step? This one. The stain, or the step where you are trying to remove the color. Because if you put, imagine, I ended up putting too much of alcohol, the color from everything will go away. Even the purple color will not stay for that long. If I put too much of alcohol, then everything, the entire slide will become blank. If I don't put too much of alcohol or the right amount of alcohol, the gram negative will not become colorless. So I can say this is a very timed and a crucial process. You can't even put it under alcohol for 2 minutes. You can't even put it up for just half a second. You have to do a proper timing. So please remember, when asked in the exam, which is the most crucial step that you have? Which is the most crucial step that you have in gram staining? And that is the step of decolorization. That is the step of decolorization. One more variation that comes in the paper. See, the first stain was crystal violet. Now, sometimes instead of crystal violet, they can add maybe something called methyl violet, or they might say gentian violet. So your concept is something violet will be written. Either crystal violet or methyl violet, crystal violet, methyl violet, or gentian violet. Any of these can be written, something violet. Obviously, what is coming to your mind next is going to be, what was the reason? Why are the end result, why is it that the gram positive organisms are going to remain purple and the gram negative organisms gave away their color, and later on the gram negative organisms became red? So please remember, right now you might have not studied the positive organisms, but let me tell you, P for P, gram positive organisms. Remember that gram positive organisms are going to contain a lot of P for peptidoglycan. Gram-positive organisms contain a lot of peptidoglycan. They have a lot of peptidoglycan. That is the reason that, for example, they have multiple layers of peptidoglycan. So, once the purple color goes inside, it stays inside. Then you can't get it out. So if you've gone too deep into some thought process, no one can get you out of it. Same way, the purple color, initially, the purple color, the crystal violet, goes so much deep into peptidoglycan, then later on no alcohol and no one can get it out of it. Therefore, once purple, always purple. Another reason is, if gram positive, if positive had P, peptidoglycan, what does gram negative have? Gram negative is going to have lipopolysaccharides. What did I write? Gram negative has lipopolysaccharides. Can I indirectly say that the gram negative organisms have too much of lipid content? The gram negative organisms have too much of lipid. Think logically. If I treat lipid with alcohol, like I did, in which step? Come in and stain. I treated it with alcohol. Will alcohol dissolve the lipid? Of course. Will alcohol dissolve all that lipid? Yes. That is why the color also came out. I put alcohol and that entire lipopolysaccharide layer, alcohol melted away like alcohol, like just melted all the fat like butter. Like all the lipid went out. That is why suddenly the color went away. Gram negative became colorless. And then which stain did I add to give it some color? I added some saffron in red. So basically I have made way for the red color stain. Entire lipopolysaccharide layer has basically gone off after alcohol. So I've given way for the second dye to come in, and that second dye is going to be saffron in red. I hope you've understood the physiology behind it. Okay, having said that, all said and done, but all throughout, I'm not going into that detail right now, but all throughout I will tell you, okay, this is gram positive, this is gram negative. But there are some things which are poorly gram staining. This is something you will find, it will be written, which are the organisms which are poorly gram staining. That is, they are little atypical. They are not like just gram positive cell wall, not exactly gram negative. They are somewhere hanging in between. So, gram is not going to be the stain for them. For them, I will tell you some other stains, but those organisms, and your mnemonic is MRCS. Which organisms are poorly staining? Like I said, MRCS, just get familiar with their names. We've got Mycoplasma, number 1. Next, we've got Rickettsia. Next, we've got Chlamydia. And next, we've got Spirochetes. Remember, when we are talking about the Mycoplasma, Rickettsia, Chlamydia, and Spirochetes, MRCS is the poorly gram staining. Having said that, First stain is done, gram staining, the bread and butter is done. Now we come to the second one. What did I tell you the mnemonic for the second one? Acid fast stain, Zn stain, that is class has asked mnemonic. The next stain that we are going to start with is the Zn stain or the Zeal-Nelson stain. So let us write the mnemonic once again over here, and that is class has asked mnemonic. The next thing is class has asked mnemonic. So first and foremost, I want to take you all the way back to the scientist section, because do you remember Zeal and Nelson were just the scientists who modified this stain. What stain am I talking about? The acid fast stain. But who was the original scientist who gave this stain to us? And the original scientist was Sir Paul Ehrlich, if you remember. Okay, coming back. What is class has asked mnemonic? So the first thing that is added is going to be a reddish color stain called carbol fuchsin. The first one is going to be carbol fuchsin. So carbol means it has some phenol element, right? So carbol fuchsin. So that is the C. The next one is H, and that is going to be heating. You can see you've put it on some apparatus to heat it. So the next one is going to be heat. The third one is A. So remember, this is again, these A and decolorizer steps are always very famous. So what are you adding over here? Usually we add acid, or we add a mixture of acid alcohol. And the most common acid used is sulfuric acid, that is H2SO4. And finally, class has asked mnemonic. So you're going to add something with M. Now it's up to you. Your second color is up to you. If you write blue color, then the stain that you are going to add is methylene blue. If you say no, I like green color, then the stain that you are going to add is going to be the famous malachite green. And let me tell you that malachite green is not very commonly used. It is not a very commonly used stain, because as per WHO recommendations, methylene blue should be used. But yeah, so repeating once again, class has asked mnemonic. The primary color that we are using is going to be carbol fuchsin. Then over here, if the mordant is asked, what is helping in that hammer technique? So if the mordant is asked, then the mordant is going to be the heating process. Then we add the decolorizer. So what is the decolorizer in this case? The decolorizer is acid alcohol. And finally, the secondary color depends. If I want blue, I put methylene blue. If I want green, I put malachite green. So now look at this final slide that I've received, guys. What are you seeing? What is the organism? If I see, the organism has come out to be red in color. And what is the color of the background? The background has come out to be blue in color. Can you correlate why? Why is the organism come out to be red in color? Because what red color stain did we use? We had used carbol fuchsin, the red color stain. And why has the background come out to be blue in color, guys? The background is blue because in the end, the stain that I used was methylene blue. All said and done. Okay, having said that, the story is not over. This was just basic. Class has asked mnemonic. Now comes the differences. How much of H2SO4 has been used? Now listen to that very carefully. How much of H2SO4 has been used? So remember, that depends. TB. I've learned TB as T20. Okay, I've learned TB as T20. So for TB, it is going to be 20%. So remember, for TB, it is T20. For lepra, so how much of 20% H2SO4? For lepra, count the number of alphabets. Lepra has 5 alphabets. So particularly for lepra, and also there is a family of parasites called the coccidian parasites. For them, we use 5% H2SO4. And let me make a table over here, which will really help you when you go to different PYQs and attempt it. In your PYQs, guys, you will come across a stain called the Fight for Acco stain. You are all going to come across the Fight for Acco stain. And what must you be knowing about it? As soon as you hear the word fight, it should remind you of 5. You know, Fight for Acco stain is the same Zn stain. It's the same Zeal-Nelson acid fast stain. Class has asked mnemonic. Same thing. But the only difference is that instead of using a 20% H2SO4, I have used a 5% H2SO4. So this is the one that is coming across as 5% H2SO4 wala Zn stain. So my easy way of learning it was that the word fight should immediately remind me of the number 5. And that is going to be responsible for staining the lepra bacillus. So when we come to the diagnosis of lepra, LEPRA, 5 alphabets, 5% H2SO4, fight for acostane, 555, okay? Even for the coccidian parasites, we use 5% H2SO4, but we don't call it fight for acostane, okay? Now, in the other order, Nocardia legionella, I'm going in the descending order. For Nocardia legionella, we use a 1% H2SO4. And for spores and sperm, S and S, how do you write an S? S is written like this, right? So ulta, make it opposite. The other one, S, is going to be like this. Something to do with, this is looking like a 5, this is looking like a 2. So all these numbers need to be taken into account. It is ranging from 0.25 to 0.5%. So let's do a very quick recap. Number 1, for TB, how did I learn? T20 matches, so 20% H2SO4. For lepra, LEPRA, 5 alphabets, 5%, fight for acostane. For Nocardia legionella, 1%, and for spore and sperm, SS1s, 0.25 to 0.5. So does that tell you, did you notice that Zn stain is positive in so many things? So your next question, what all is Zn stain going to be positive in? So now guys, there's a mnemonic for it, and every mnemonic that I'm trying to give you one, like for every scientist, we did it with the scientist's name. For gram staining, I told you, you have to learn it. So come in the lab and learn it. So we are trying to get some orientation around it. So what are the acid fast things? For the acid fast, I'm going to tell you my nose is cold and hot. But now you will ask me, why are you telling me? What sense is this making? So first, let me make sense to the mnemonic. There is a modification of the Zn stain known as the Kinyon stain. Now, what do I mean by that? See, when I say Kinyon, Kinyon is sounding like a K. So this means this is what modification? This is cold Zn stain. Have you ever wondered what is cold? Am I going to freeze it? No, not at all. Tell me, what was the mnemonic for Zn stain? What were you doing in Zn stain? You were saying there is class has asked mnemonic. Can you repeat with me what has happened? C was for carbonyl fusion, H was for heating, A was for H2SO4 acid, and M was for methylene blue. Now I'm saying I'm doing a cold Zn stain. So what are you just going to remove? Which step will be removed? The heating process is removed. The heating process is removed. But does that not make any difference? If it was so casual, then why did we have the heating process? What was heat doing? Heat was that hammer. Heat was the hammer or the mordant. Now there is no hammer. What do? Then I will have to do it something in the first step only. I will have to modify so that the heating can be skipped. So remember, I increase the concentration of phenol in carbonyl fusion. So remember, in that C, carbonyl fusion, I told you what is carbonyl. Carbonyl is a phenol. So I increase the concentration of phenol because I know later on no hammer is going to come. So let me add a lot of phenol in the beginning only. Okay, having said that, when would I do this situation? I do it for the diagnosis of a family of parasites called coccidian parasites. Now guys, please remember, how am I going to learn all of this? So I've learned it as a KKK sound, okay? So what is it? The first thing that I'm talking about is the Kinyon stain. Kinyon stain is heated wala stain or cold wala stain. It is a K for cold Zn stain. And for which family are you using it? I am using it for a coccidian parasites. Repeating, K for Kinyon, cold, coccidian, cold, coccidian, Kinyon. And there is a recent question on, is there any other stain for Kinyon or other name for Kinyon? This is also known as the Gabbett stain. You will find this in your previous year questions. This is also known as the Gabbett stain. Okay, all said and done. Now did you analyze? Why did I make my mnemonic? What are all the things which are Zn stain positive? I said my nose is cold and hot. Why did I make this cold and hot mnemonic? Because yeah, Zn is all about heating and then not heating, all about hot and cold. So now tell me, what are all the things which are Zn positive? So my stands for all the mycobacteria. When I talk about mycobacteria, what all mycobacteria come to your mind? TB comes to my mind, atypical TB also comes to my mind, and leprosy also comes to my mind. But do I know how to differentiate between them? All of them are Zn positive. I think I know. When I'm talking about TB and TB, be it typical TB or atypical TB, do I know they are acid fast with how much of H2SO4? They are T20, so they are acid fast with 20% H2SO4. But then how did I differentiate between them? Because I realized that both of them are acid fast. Both of them are going to be acid fast. So difference kya hai? TB is also alcohol fast. means if you treat it, how much of alcohol? TB can also be alcohol fast, whereas atypical TB is non-alcohol fast or just acid fast is how you can learn it as well. So repeating once again, guys, listen to me very, very carefully. TB is both. I'm going to write this once again because I want it to be as crystal clear as possible, because this concept will not come again. Please remember, typical TB is both acid and alcohol fast, whereas atypical TB is only and only acid fast. So you were asking me the concentrations, na? Acid ka concentration har jagah kitna hota hai? How much of H2SO4 do we use? T20, we always use 20%. If they ask you the concentration of alcohol, they usually use a 95% alcohol is used, okay? 95% alcohol. So simple is that if both of them are coming acid fast, if something is coming acid fast, you want to find out is it typical TB or atypical TB, do an alcohol fast staining. If you will get something which is alcohol fast, you will get to know that, yeah, this is typical TB. So typical TB is total. That is how I've learnt it. Now full mnemonic. Typical TB is total. Acid fast also, alcohol fast also. Next, if you wanted to differentiate lepra, I think the acid content is only clear to you. Lepra, 5 alphabets. How much of H2SO4 will it be acid fast with? It will be acid fast with 5% H2SO4. I hope the concept is clear. I'm moving on to the next. So my nose. No is going to be nocardia and legionella. Nocardia is NO, and also along with that I want you to learn legionella. So my nose. Okay, what is with I? Now I stands for isospora. I stands for isospora. Okay, I'm not talking about the next S I'm going to come to in a while. But which family is isospora? You will eventually read it in the parasitology section. Isospora has a cousin brother called cyclospora. Isospora has a cousin brother called cyclospora, and they also have a cousin brother that is cryptosporidium. They also have a cousin brother called cryptosporidium. And do you know what three of them, why am I calling them cousin brothers? The spora brothers, isospora, cyclospora and cryptosporidium, they fall under the coccidian parasites. These three members are members of the coccidian parasites. So tell me something, guys. Are you going to use the regular Zn stain or are you going to use the CO cold Zn stain? I will prefer the cold Zn stain, hai na? Okay, the S was still missing. So I for isospora, C for cyclospora, cryptosporidium. The S is going to be number 1 for spores. And next is the head of the sperm, because right now I just taught you how much percentage of H2SO4 will you use for S for spores and S for sperms? 0.25 to 0.5%. Hai na? Very good. Now coming to the next one. My nose is cold and hot. So this hot also we will have to split it up. We will have to split it up into HOT. So that means H stands for hooklets. Remember, hooklets of hydated. Hooklets of hydated cyst, they are acid fast, and the eggs of Tinea saginata. The eggs of Tinea saginata, they can also be. So now T se matlab tinea. Kaun sa tinea? There are two tineas. Tinea solium. Solium doesn't have a T. Saginata has a T. So Tinea saginata is going to be acid fast. It's quite a lengthy and a stressful mnemonic, but at least it covers up everything from path and micro that you have to know for Zn stain. Let's do a very rapid review. My stands for all mycobacteria. TB, atypical TB, leprosy. No for nocardia, legionella. I and C for isospora, cyclospora, cryptosporidium. S is going to be for spores and sperm. And hot ka HO is going to be the hooklet. And T is going to be the egg of which? Tinea, Tinea saginata. These are all the things which are Zn stain positive. Which brings us back. Which was the first stain that we did? Gram. The second one was the Zn or the acid fast. And the third one is going to be the Albert stain. Very important previous year question that we all should be knowing. So when we talk about the Albert stain, first I'll have to tell you what is it with Albert stain that you're trying to do. So please remember, right now you've not done bacteriology, but I'm sure eventually you will. Albert stain is used for something known as volutin granules. Okay, Albert stain is used for volutin granules. Do you want to know what are volutin granules? Volutin granules are present in a bacteria called Corynebacterium diphtheriae. Volutin granules are positive in Corynebacterium diphtheriae. So I'll try to draw it. So see, what have I drawn over here? I have drawn this as Corynebacterium diphtheria. Repeating once again, this is the Corynebacterium diphtheria. And I am seeing that at the two poles, I am seeing some granules. So these are the volutin granules. These are the volutin granules. Is this the only name that they have? No. Volutin granules are also going to have some other fancy names. This was just one of them. The other fancy name is known as Mbbs granules. These are also known as Mbbs granules. Now can you see where are they? They are at the two poles. One is over here, right? And one is over here. So if they are at the two poles, can I call them bipolar granules? I am also going to call them bipolar granules. And finally, the last name given to them are metachromatic granules. So now I think I've learnt most of the names. See, volutin I have understood, and Mbbs, these must be some fancy scientist and places wala naam. Bipolar I have understood because it is present only at the two poles, so that also I've understood. Metachromatic word I have to understand. Metachroma. Meta means something different. Chroma means color. So what is the concept? For that I'll have to teach you what is the stain for volutin granules. Now please remember, the first and foremost, I'm going to move on to a separate slide first. Please remember, the stain for volutin granules is we are making a plan. Okay, we are making a plan. So what is we? We for obviously volutin granules, and we are making a plan means that there are these 4 stains that you have to know. One is going to be the Ponders stain. The next is going to be Loeffler methylene blue. Loeffler methylene blue. The next is going to be Albert stain. And the next one is going to be the Neisser stain. The next one is Neisser stain. So having said that, what do we have for plan? Repeating once again, we have Ponders stain, Loeffler methylene blue, Albert stain and Neisser stain. Out of them, now the latest question is that Loeffler's methylene blue is considered as the best. That's a PYQ that you've been getting. But what you actually get an image on is the Albert stain, and that is exactly why I had drawn that picture right now. The picture that I had drawn was a picture of Albert stain. So I'm going to come back and show you. The picture that you're seeing over here is the picture of which stain? Albert. So how many stains are there for volutin granules? Ponders, Loeffler methylene blue, Albert, Neisser. But which one am I showing you here? I'm showing you the Albert stain. Albert stain has 2 solutions. Albert 1, Albert 2. Albert 1 and Albert 2. So Albert 1 has something. Acha, did you notice something, guys? What are the 2 colors that I'm using here? For the organism, I have used a green color. This means I have to have something greenish in my stain. And for the volutin granules, I've used some bluish purple color. So this means I'm supposed to have some bluish purple stain. That solves your purpose. I need a blue and I need a green. So I've added toluidine blue for blue color and malachite green for green color. Little bit of acid which you can add anywhere. That is Albert. So Albert 1 is going to have all the colors, the blue and the green. Albert 2 is going to have iodine, only and only iodine in it. So now the examiner will ask you that what is the color that the organism has got? The organism has got a green color. And why has it got a green color? Because I have used a stain called malachite green. What is the color, guys, that this volutin granules have got? So now the twist in the story is that volutin granules have got a purple color. But now why is that a twist in the story? Because the stain that I used was a blue color stain. The stain that I used was blue in color, so I should have got a blue color. But the color that I'm getting in return is a purple color. All of us, this concept, it has a different color. So if I give you a bottle of toluidine blue, it will be blue in color. But when you look at it under the microscope, it's going to be purple in color. So two-faced. Some color, it's like everyone has that one topper in class who says, I have not studied anything, but then on the day of the exam, you see in the viva, he or she is talking non-stop and writing down on hundreds of continuation sheets. Yes, we've all known that topper once in our lifetime. Same way. Toluidine blue is that topper. Toluidine blue has what color? It has a blue color. But what color does it give to the slide? It gives a purple color. Separate color, but giving separate color. This concept is known as metachromasia. This is a metachromatic stain. What is metachromatic? Metachromatic is girgit or chameleon. That means it has a different color and it will give a different color. That is why I call these granules metachromatic granules also, because they are stained by a metachromatic stain called toluidine blue. So either you'll get a question, arrange the order. So you'll say first simplest, I hope I get this question. First you'll put Albert solution 1, then you'll put Albert solution 2. Sometimes you might get a question where all these things are written separately. You have one bottle of toluidine blue, one bottle of malachite green, one bottle of iodine. In that situation, if it arises, follow the mnemonic TIM. And what is TIM? First, what will you add? Toluidine blue, then iodine, and then malachite green. Okay, so just in case they are split up, you use the mnemonic TIM. If they are given as Albert 1 and Albert 2, no doubt you put first and then you put the second one. And that wraps up everything, but I want to tell you something extra. And that is, are these volutin granules only seen over here? No. Please remember, the way you write volutin, you are supposed to take first alphabet, last alphabet. Then you say granules, over there also you are supposed to take the first alphabet and the last alphabet. So that means that where all, firstly I told you volutin granules are the mainstay of Corynebacterium diphtheria, but apart from Corynebacterium diphtheria, they are seen in Spirillum, they are seen in Gardnerella, they are seen in V for, now we are going to change it a little, V ko change karenge into a Y, right? And ye jo N hai, this N will be changed into an M. These two changes you will have to do, because where else do you see it? That first and foremost, Y for, it is seen in Yersinia pestis. It is seen in Yersinia pestis. It can also be seen in certain yeast forms. And YM, because this is also seen in Mycobacterium TB. So I hope that you will remember the mnemonic, that the mnemonic for the volutin granules is going to be the volutin granules only, which means you take the first alphabet V, you change it into a Y, so that is yeast and Yersinia. N is changed to M, so that is Mycobacterium TB. G is Gardnerella, S is Spirillum. And of course, Corynebacterium diphtheria, you cannot ignore, that will pakka be there. Okay, that finishes off our entire story of these volutin granules. But yes, almost our stains have come to an end. The last thing that I want to tell you are two miscellaneous stains, stains for spores and stains for flagella. So let me quickly tell you flagella. Flagella is something which is a very, very thin structure, very, very thin structure. Right now, I told you that if you want to put up flagella, which stain can you use? Can I also use the silver stains, the impregnation method? Remember, I told you that if there's a very, very thin structure, you're going to make it fat by adding a lot of things on it, yes. But other than that, we have something known as the Leifson and Reuss stain. If you're feeling worried that am I supposed to learn everything, don't worry. What have they said? That the background of the cell is going to become what? The background of the cell has become something bluish. And what is the color of the flagella? The color of the flagella that I have used is something reddish. That's all that you have to know. But you know, actually, honestly telling you, what is the question that's going to come? Flagella ka red color kyun hai? Because you have used fustian. That's all firstly that you have to know. Why is flagella red in color? Because whenever I use fustian, that is going to give me a red color. Question number 1. Question number 2 is this, what has come so many times. What is the Leifson and Reuss stain that is going to be the mordant in Leifson and Reuss stain? So in Leifson and Reuss stain, the mordant is tannic acid. How do I learn this information? Never mind. We will make mnemonics because that is the only thing that is going to save us. What does flagella help in? Flagella helps in locomotion. So something helping in locomotion is going to have what stain? It is going to have the Leifson-Reuss stain. It is going to have the Leifson-Reuss stain. In the Leifson-Reuss stain, why is the flagella looking red in color? Why is it looking red in color? Because we have added fustian to it. That is why it is looking red in color. So simple. F for flagella ho gaya, F for fustian ho gaya, red in color. It helps in locomotion. So the name of the stain is the Leifson and the Reuss stain. And the last question is what is the mordant, which I think everyone will know, that is tannic acid. Having said that, the last thing that you need to know is to do with the spores. What are the stains of the spores? S for S. S for S means it is the Schaffer and Fulton stain, also known as modified Ashby stain. Before that, what is a spore? That you should know, because you've not studied bacteriology right now. Spore is nothing but the resting form. It's the resting or the dormant form. It's the resting or the dormant form that you have of a bacteria. So when the bacteria wants to rest a little, it's going to form a chota sa spore and it's going to go off to sleep. Now, tell me one thing, guys. When you go off to sleep in your house, and a very common Indian household thing that I'm going to share, and you'll relate to it definitely. Whenever you go off to sleep at odd timings in your house, like you wake up at say late 11 o'clock in the morning, or you wake up at 2 in the afternoon, right? So how do your house people address you? Mr. So-and-so has woken up, as a taunt, or Miss Queen Victoria has woken up, or the king has woken up, or the princess has woken up. Have you heard these taunts? And this is a very nice way of saying it. In Indian households, there are many other crude ways of saying this also. But spore is the sleeping form. So now, because it is sleeping, I am becoming like an Indian mother and I'm giving it a little bit of taunts. I am calling it, oh, Miss Spore has finally slept off and is waking up. Miss Spore. That is why I'm calling it Miss Spore. Why did I say Miss Spore? Because now I want to tell you what are the two stains that I used in spore. One is going to be Miss, M and S. So one is going to be malachite green and the other is going to be saffron in red. So simple. What is Miss Spore? M is going to be for malachite green and S is going to be for saffron in red. Now, having said that, please remember and look into this picture carefully. Can you all see how the organism has come out to be red? Whatever bacteria we had has come out to be red. Why is it coming out to be red? Because Miss, which is the red one, I have used saffron in red for this. And what is the spore? Figure out the spore. Yes, I can see these green color round sleeping organisms. These must have been the malachite green. So now that is why I called it Miss Spore, because I realized that the bacteria is coming out to be red. The bacteria is coming out to be red. So that is saffron in red. And the spore is coming out to be green. And that is why I called it malachite green. I hope everyone is okay. So as soon as spore comes, you know S for S. For spore, you have the Schaffer and Fulton stain. And you're going to address it like a typical Indian mother trying to taunt that, oh, Miss Spore is sleeping in the house. So malachite green and saffron in red. That finishes off not only the scientists but also the stains. I think it's time that we pick up a few questions. So let's start our discussion with this question over here. In the gram staining, what is the mordant? Okay. So now, whenever you get these questions, make it a habit that you write down the mnemonic or you think of the mnemonic. Come in and stain. So what was the mordant? If you remember, come in, what was I? I was for Lugol's iodine. Lugol's iodine was the... So let's do a quick recap. What was C? C was for the violet. So it was crystal violet. After that, you had I for iodine. Then A was for some alcohol or acetone. And S was for, in the end, saffron in red. So mordant was iodine. Let's pick up the next question, which you need to read very carefully. Very interesting question. After the primary stain and the mordant has been added, but before the decolorizer agent is used, gram-positive organisms are dash and gram-negative organisms are dash. So see, all of us are very used to doing the retrospective approach. If I would have read only half the line and said gram-positive organisms are dash, gram-negative are dash, I would have said, okay, gram-positive are going to be purple and gram-negative are going to be pink or red. I would have marked as C, but that's not the right way of approaching this. They have given me where exactly do you have to pick it up. So let me write. Come in and stain. They have said the primary stain has been added, the mordant has been added. Right now, all organisms are what in color? I had studied that when crystal violet will be added and iodine will be added, at that moment, everything is going to be purple in color, whether it is gram negative or it is gram positive. So right now, everything is purple. But before the decolorizing, so they have stopped the process here. They have not added the decolorizer. Right now, what is the stain color of all the organisms? Right now, everything is purple. So right now, gram positive is also purple and gram negative is also purple. If I would have added the decolorizer, then the story would have changed. But here they've mentioned before the decolorizer has been used. Having said that, I hope that is clear. Let's move on to the next question. Metachromatic granules are stained by all except. Do you remember metachromatic granules were nothing but the volutin granules? And I told you that for the volutin granules, we basically need a plan. And what is the plan? Ponders, Loeffler, Albert, Neisser. Ponder, Loeffler's, Albert, Neisser. All of these are used. What is not used is Quignon, because what was Quignon? Quignon was the cold Zn stain. It has nothing to do with volutin or metachromatic granules. So which one is not used? Option D. Coming to the next question. Silver impregnation method of staining is used to demonstrate what? Silver staining. So you have mycobacteria, spirochetes, both or none. Whenever these kind of both questions come, both of the above, all of the above, it's a general temptation that you want to mark it, but don't fall for that trap. Please remember, when I'm talking about silver, I have to think of thin structures. So I'll be thinking of structures like spirochetes, flagella, thin structures, silver impregnation. Coming on to another question. Which of the following are acid fast with 20% H2SO4, T20? Yes, guys, what is T20? Some TB, mycobacteria. So TB, that could be typical, that could be atypical, right? So TB. So what is it? Which of them? Mycobacterium avium. You're not seeing TB here, but Mycobacterium avium is an atypical TB. Can you also, recall what was for lepra? Lepra 5 alphabets. So how much percentage was there? 5% H2SO4. But for TB, T20. Coming on to the next question. In the Zn staining procedure, the secondary stain is, first, remember your mnemonic. Class has asked mnemonic, in which C was for carbonyl fusion, H was for heat, A was for acid, that is sulfuric acid, and M, I told you if you like blue color, you can select methylene blue. And I said if you like a green color, you can also select malachite green. But methylene blue was the famous one. Do I have? So now they are asking you what? They are asking you secondary stain. So matlab, primary color was carbonyl fusion. Secondary color was methylene blue. Do I have methylene blue written anywhere? Yes. So that becomes the answer for you. So for these kind of questions, think of your answer and search for it in the options. Coming on to the last question that we have, and then we'll wrap up this segment. What is the correct order of gram staining? Again, think of your answer. Come in and stain. Crystal violet I have to find for some violet. So I have lots of violets, crystal violet, methyl violet. So all of them will work, some violet. But after that, immediately I need iodine. That is being fulfilled only in one option. Crystal violet, iodine, then either alcohol or acetone, that is also correct. And fourth one, safranin, that is also correct. So this option B is fitting very well in my answer. So that's the final answer over here. With that, we wrap up our discussion. And now we are going to start with the next topic, which is going to be the microscopes.